Hello, and how are you? Welcome to this session on meiosis. My name is Winnie Barawa. We have begun with mitosis, and if you've not watched that video, you can link up on our playlist and be able to check because this is a carry-on of what happened in mitosis, or rather, it's a more so like a similar replica of what happens in mitosis. So let's keep on and see what happens. So by introduction, meiosis is the process of cell division where one cell with diploid number of chromosomes will divide twice to form four cells, each with a haploid number of chromosomes. And in humans, we expect 23 chromosomes at the end of the meiosis process. So it happens in two processes. It can happen as what we call oogenesis, and this is happening in the ovaries to form the egg cells or the ovum, while in sperms we call it spermatogenesis that happens in the testes to help us form the sperm cells. However, during fertilization of the ovum by the sperm, we are able to restore the diploid number of the cell of the fertilized eggs, being reported that all gametes are haploid in nature, meaning they contain half the number of the mother cell. So how summary can we talk about meiosis? So in meiosis, we are saying this is a cell division process where a diploid number of chromosome will divide twice to produce four daughter cells that have half of the original parent cell chromosomal number. So if we started with 46, we will end up with 23 on each of the daughter cells. So first and foremost, before we go into details how the meiosis process happens, I need us to understand a few concepts that pertains to meiosis. And part of it is understanding what sister chromatids means. And sister chromatids, these are two halves of a duplicated chromosome, okay? And then we have homologous chromosomes, which refers to similar but non-identical chromosomal pairs. So let's start with these strands here. They look like strands, but actually these are chromosomes. So the single strand here, we are calling it chromosome, and we are calling them homologous chromosomes. So what homologous chromosomes have, they have the ability to replicate and produce a complementary copy of itself. So when homologous chromosomes replicate, they can produce a sister. So let's use the green here. So our green will replicate and produce a sister copy. So the sister copy will be attached at an area here that you are calling centromeres. So these sisters, we are going to call them now sister chromatids. So we shall not call them homologous chromosomes. Yes, they are originally homologous chromosomes, but they are now existing as sister chromatids. So we have two sister chromatids here. One is purple and one is green. I need us to understand this concept because it's going to guide us during the meiosis division. And the other concepts are the concept about synapses. And in synapses, it's whereby there's pairing of the homologous chromosomes to form a tetrad. So we said when the homologous chromosomes replicate, they give us sister chromatids. But in a normal person, sister chromatids are partly from the father and partly from the mother, 50-50. So you have maternal sister chromatids and you have paternal sister chromatids. When these chromatids come together to pair, they will give you what we call a tetrad. This is like four chromatids here together. So pairing of homologous chromosomes, to form a tetrad. Then on the other side, tetrad is just a definition that these are four sister chromatids of the paired homologous chromosomes. I hope that makes it clear. And then you have a process called crossing over. So crossing over is the state whereby we can have at some time a recombination of the alleles in our homologous chromosome. An allele is a section of the chromosome that carries our genetic composition or our genetic characteristics. So what happens with crossing over is that part of this allele or genetic or or segment can be crossed over, or it can be broken up and patched into another part of a chromosome, such that there's exchange of genetic materials or exchange of genetic information. So crossing over can be defined as sharing of genetic materials between two non-sister chromatids in a homologous pair. So here we have our homologous chromosomes that have replicated, and it has created sister chromatids. So these tetrads here, what can happen is they can do crossing over. So one can exchange material with the other person. So the exchange of this material here, this part where we are crossing over, we call it synapsis, and the place where we touch, the touching parts, we call it the chiasma. At the end of the day, we end up with recombinant chromatids. Recombinant chromatids here we are referring to the chromatids that exchange genetic materials. However, we have chromatids that didn't exchange materials, and these chromatids we are calling them the non-recombinant chromatids. Okay, so in summary, we are saying in meiosis, you have to understand about homologous pairs, synapses, and the crossing over. The crossing over, by summary, we are saying this is the exchange of genetic material between the chromatids in the tetrad. And in the exchange of this material, we create the synapses. And once the material has successfully happened, we describe that process as crossing over. And the point where the exchange of this genetic material successfully happens, that place we call it chiasma. Chiasma. So we end up with the recombinant chromatids and the non-recombinant chromatids. This is a bivalent. Bivalent means you have two pairs of homologous chromosomes that are generally creating a tetrad for you. So let's describe meiosis now in its required phases. So meiosis happens in two phases, where we have meiosis 1 and then we have meiosis 2. So because this is part of cell division, it falls under mitotic phase, only that this happens a bit differently, where the original cell will divide twice or two times to give you a haploid daughter cell. So this is the mother cell. I just want us to use this example to elaborate on this. So this is the mother cell. And what happens with this mother cell during cell life cycle? There will be the replication. So it will replicate to produce other sister chromatids. So originally you can see the chromatids here, there are 4. Now the chromatids here are now 8. So in the division of the meiosis, we shall have meiosis 1 and meiosis 2. In meiosis 1, we talk about division of the homologous chromosomes into equal number. So if there are 4, we shall divide the homologous chromosomes into 2. So we shall have 2 going on the other side and 2 going on the other side. Then on the meiosis 2, this is division of the chromatids, or the sister chromatids. So we shall divide these chromatids here into 2. So we shall have chromosomes that are half the original mother cell. Are you seeing now? So there were 4. Here we are ending up with 2. So keep up with me. We want to explain this now step by step and how it happens. So what happens in meiosis 1? What happens in meiosis 2? So in the meiosis phases, we are saying we have meiosis 1 and then we have meiosis 2. So in meiosis 1, we say that the cell will go through prophase 1, metaphase 1, anaphase 1, telophase 1 together with cytokinosis. Then this cell will transit to the second stage of meiosis division that we are calling meiosis 2. So in the meiosis 2 division, we shall have prophase 2, metaphase 2, anaphase 2, telophase 2 together with cytokinosis, and at the end of the day we will have 4 daughter cells with half the chromosomal number from the mother cell. So there'll be haploid cells. So we have mentioned that meiosis 1 is the first round of the cell division. So in meiosis 1, there is separation of the homologous chromosome or separation of the homologous pair of chromosome during this first round, and it will go through prophase 1, metaphase 1, anaphase 1, and telophase 1. So what happens in these phases? In prophase 1, the chromosome will begin to condense and pair up through a process we call synapsis and allow exchange of the genetic material, and this process we are calling it crossing over. So it is at this stage that also the nuclear membrane breaks down. So in prophase 1 of meiosis 1, we have prophase 1 where the chromosomes are condensing, they are pairing up through synapses, and we are allowing exchange of genetic material that we are calling alleles through a process we call crossing over. But now the membrane, the nucleus membrane also is breaking down and disintegrating. And after prophase 1, the cell will go through metaphase 1. So in the metaphase 1 phase, each single chromosome will now attach to the microtubule, just one pole of the spindle, and they will align at the equator to form the metaphase plate. So as you can see, the procedures are literally like what is happening in mitosis, but in this case, we are focusing on the homologous chromosomes in meiosis 1. So we are not separating the chromatids, we are separating only the chromosomes. I will demonstrate this again. So in metaphase 1, now the chromosomes are aligning at the equator, and each microtubule will be attaching to the chromosome. And then at anaphase 1 now, we have the homologous chromosomes will be pulled apart into the two opposite poles. However, I repeat, the sister chromatids will remain attached. And the last phase is telophase 1, where the chromosomes have arrived at the opposite poles. The nuclear membrane is reforming, and the chromosomes will condense and cytokinesis will begin. So when cytokinesis will begin, we will have produced two daughter cells, which are still diploid, but they will go through the second phase of division that we are calling meiosis 2, that will give us now the haploid daughter cells. So this is what we mean when we are saying we are separating the homologous chromosomes only. So this is our cell that has completed the interphase and the DNA replication has taken place. So we are headed to prophase 1 stage. So in this case, the homologous chromosomes are condensing and becoming visible, and the centrioles are moving to the opposite poles while producing the spindle fibers that are targeting to attach on these homologous chromosomes. So at the metaphase stage, the homologous pair of chromosomes are now attached or have aligned at the metaphase plate by aligning at the equator. However, like I said, the spindle fibers will be attaching to each homologous chromosomes as opposed to the chromatids. So it will attach as each spindle will be attaching a single homologous chromosome, such that during anaphase we shall separate the homologous chromosomes into two separate ends. So we shall be separating the homologous chromosomes while the sister chromatids stay intact. So can you see? So this is a homologue, and the homologue is now moving into the opposite direction, but the sister chromatids are intact. So by the end of the telophase, we have newly formed cells. Okay, these are not yet haploid. They have still chromatids, and the chromosomal number is more like identical to the parental chromosome. So the second phase now is what we call meiosis 2. And it is in meiosis 2 that we shall now complete our meiosis process. So in meiosis 2, this is now separation. I want you to note in meiosis 2, we shall go through separation of the sister chromatids during the second round, and they are taking over from the cells that have gone through meiosis 1. So in this stage, we shall have prophase 2, metaphase 2, anaphase 2, and telophase 2. And then cytokinesis will happen again, and we shall split our cells into 4 new haploid cells. So in prophase 2, the chromosomes again will do the condensing. So the chromosomes will condense and then the nuclear envelope will break down. So the centrosomes will move towards the opposite pole, and that is the north and the south pole. Then in metaphase 2 phase, what happens is the chromosome will line up along the metaphase plate or along the equator to form the metaphase phase, and the microtubules will now attach to each sister chromosome, sister chromatid of each chromosome. So we shall attach this microtubule at the kinetomere to just help us in the cell division. So at anaphase 2, the sister chromatids will now separate, and we shall use the spindle fibers or the microtubules to pull them apart towards the opposite poles of the cell. And in telophase 2, the nuclear membrane will start to form around each cell of chromosomes, and the chromosomes will now decondense to help us create our nucleolus materials. And the cytokinesis now will split this cell into the 4 new haploid cells. So this is what happens in meiosis 2. We are saying these are the 2 daughter cells that have completed meiosis 1, and we have these cells starting over at prophase 2. So the 2 cells at prophase 2, they will decondense, have the centromere producing the spindle fibers, and they will come attached at the sister chromatids now. So the spindles will attach at each sister chromatids during the metaphase and form the metaphase plate. And then during anaphase, we shall separate the sister chromatids. And once this successfully happens in telophase, we shall have formed new daughter cells with haploid state. That is, they'll have half the chromosomal number like the mother cell or the parental cell. So thank you so much for being with me all the way to the end pertaining mitosis and meiosis. Don't miss the next class as we're going to explore more pertaining what is the difference between mitosis and meiosis. Remember to subscribe, share these videos, and also leave us a like. Thank you so much. See you in our next class.